or Bob Wheeler. He is director of the Dental DNA Lab in Colorado Springs. He's actually a high school science teacher, and he helped develop this with the Honorable late Hal Huggins on identifying the organisms that are present in the oral cavity, and it is a footprint. It is a fingerprint of the organisms that are present. So you're not growing a culture, you're not trying to find something crazy, you're just identifying. You're taking a biopsy of the area and identifying the organisms present. And I'm going to give it over to Bob because he explains it a heck of a lot better. And do understand, he is going to pronounce some words that you and I could never get out of our lips.
Thank you, Dr. Margolis. All righty. So I'm Bob Wheeler, and I've told some of you at lunch and private meetings, I guess, about how I got into this. As Michael mentioned, I taught high school for 24 years. I retired 2 years ago. And my wife is an assistant principal at my high school. And teachers, you know, I would be there by 7 o'clock in the morning. I'd leave by 3:30, 4 p.m., depending on who needed help. I'd come home, pick up a nice book, lie down on the couch, be reading the book. My wife, being assistant principal, would come in a couple hours later, and I would ask that fateful question, honey, what's for dinner? In hindsight, that was probably not a great question to ask, because I'm not sure how many times I asked that, she said, you need to get a part-time job. And I said, okay. So I thought, well, Starbucks, Home Depot, do you need a blue tag for that? And I decided, well, I didn't want to work too hard, but I'd kind of like to use my degree in microbiology, and I had an interest in DNA. So I went online and checked, you know, a great website for looking for jobs, Craigslist. I didn't want to work too hard. And posted 3 days before, looking for a retired microbiologist with an interest in DNA. And I thought, oh man, I think I just found a job. So I went in and interviewed. It turned out to be with Dr. Huggins. He was trying to pronounce Pseudomonas aeruginosa. And I'm pretty good with it because I'd helped develop a flagellar vaccine for it when I was working at Shriners Burns Institute in Cincinnati. And I guess I impressed him. And he was funny because he said, well, you've probably heard of me. And I said, nope. And he said, well, I'm a holistic, you know, biological dentist. And I said, uh-huh, what's that? Dentist. I heard dentist. Okay. And anyway, the rest kind of became history. And this started about 5 years ago. So title of the talk, Dental DNA. These are Pine Creek, my high school's colors, by the way. Dental DNA, or detection of microbial DNA / RNA in a clinical specimen, how it works, and what do I do next? So Dr. Huggins said, Bob, I need you to design a lab. You can have the break room, it's 10 by 20 feet. I need it to be a PCR clean room. Need you to order all the equipment. Need you to do it for this amount. So I found the contractor, had the lab built, bought the equipment, designed the tests, ran the tests, explained the tests, and you know, it's kind of what I was doing after school for about 3 years before I retired from teaching. So in the beginning, a small break room. This is Rocco, my contractor. First thing we did, we had to put in a door with mesh glass to keep terrorists out. I don't know. A seamless floor had to be installed. That was kind of expensive. There's Rocco putting in our sink with our emergency eye wash fountain. The best cabinets that Home Depot had to offer. Power strips. You can never have too many power strips. Countertops. I would have picked black or white, but Jamie and other people in the office picked the countertops. So I was like, okay. And here's our first set of Eppendorf micropipetters, which you might have seen in shows like CSI. We had one power supply. Dr. Huggins had old lab equipment. That's a 15 mil centrifuge on the right and a 3-place digital balance on the left, which is much better than anything we had in high school. Had to have positive air pressure to blow the bugs out, not let them in. There's our first thermal cycler from Bulldog Bio. Single gel UV transilluminator. Had to have a -20 freezer. The microwave was not for making coffee. You really didn't want to do that because we were making agarose gels and other reagents with neurotoxic chemicals in them like tris. And the lab today, we have our name on the window now. And that's Lena doing some sample prep next to our PCR hood. That's Leslie, my PhD in molecular genetics from Hawaii, whose first comment was, "God, I look so short." And I looked at her and said, "Darling, you are." Larger centrifuge, we're spinning down our cheek cells, urine, and genetically engineered E. coli. And we went on eBay and found a 6-gel UV transilluminator so we could look at 6 gels at one time instead of one at a time for cheaper than the original one. eBay's awesome. We do wear a face shield, but we had one of my daughter's boyfriends actually, pre-med, had, wearing the safety goggles. Very, very fair. He got sunburned. So we had to actually have him put on sunscreen before he looked at gels. I don't have to. Now we're up to 3 power supplies, 10 gel electrophoresis chambers for looking at the DNA bands, 3 thermal cyclers, and we're thinking we might have to expand. Our PCR hood. Any questions? Can we go now? No, I'm sorry, I'm just getting started. You sound like my students. Oh, wait, no, you have to say, chemistry's hard. Okay, design the test. I asked Dr. Huggins, so what microbes do you want me to look for? And his response was, I don't know, you pick them out. Not helpful. So he did mention N-Haphagy and from the Forsyth Institute. So I found this one excellent paper where she described 1,000 different microbes in percent population of their occurrence in the oral cavity. So I picked the 90 that were most prevalent and we thought were most relevant, but our test is changing. So we have people asking, hey, can you send us a list of the bugs you test for? Well, I can tell you what we were testing for last month, but it changes because we have people say, can you test for this? Yeah. For example, we had a patient who had mandibular cancer, had half of her jaw scraped out because she had tested positive for blastomyces dermatitis, which is found on the fur of cats, and was concerned that she had it infected again. And I could tell her she did not. She was safe from that, and I actually met her at a conference in Chicago and got a hug. Entamoeba species, courtesy of Eric Davis from Australia, hepatitis B, chlamydia pneumoniae, Dr. Huggins, cytomegalovirus, CMV, Epstein-Barr, which causes, what they call it, infectious mononucleosis, that they say once you get it, it just never goes away, your energy levels never come back. I had the most pronounced case CSU had seen in 20 years. I can attest to that. How do we do it? Well, we use something called PCR, which stands for polymerase chain reaction, which is a standard in molecular biology, has been for about 30 years. It's now mentioned in 95% of all scientific papers written in medicine and the biological sciences. It's basically a Xerox machine. Oh, that's probably a copyright infringement. It's a Xerox machine for genes. You can take one copy and in an hour and a half have 1 billion copies. DNA fingerprinting for microbes. Instead of looking for, a crime scene, we're looking at microbes, which is kind of a crime scene. It's highly specific for our target DNA. Its specificity is 4 to the 30th power. If you punch that into a calculator, you'll get an overflow stack error, which means your calculator can't count that high. But if you Google it and type in 4 to the 30th power, it'll bring up something like 1 times 10 to the negative 18th power. It's in the quintillion, so our specificity is comparable to finding 1 grain of sand on the planet. Highly sensitive, we can detect 1 copy of DNA. In reality, that's between 1 and 10 microbes. So pretty good. We did have Clea ask, well, why are you doing PCR? Why aren't you culturing these bugs instead? And I said, well, a lot of them are obligate anaerobes, and they can take 8 weeks to grow. And for some reason, our patients get a little cranky if you make them wait 2 months to get results, when we can actually have the result in, well, we like to say 5 days, but if we're not busy, we can do it in a day. So, how does PCR work? This is courtesy of the DNA Learning Center at Cold Spring Harbor Labs, Long Island, New York. And Susan made this. So basically, you take your DNA, heat it to about 95 degrees Celsius, and you have this chunk in maybe 3 million, 6 million bases. And you say, I want this gene. So you anneal the primers. And the primers are little short segments of DNA, which are at the beginning and the end of a gene. And you have to get them so that they run this way and not this way. Otherwise, you don't amplify your gene and you get nothing. Annealing temperatures, we actually test between 45 to 70 degrees Celsius. Is to find the best temperature where the DNA will stick specifically to that target DNA and make just the one you want and not something non-specific. So in go your primers. Extend your primers with something called Taq polymerase, which stands for, well, it used to be Thermophilus aquaticus, and then Berge's manual changed it to Thermus aquaticus to sell another book. 72 degrees, denature the DNA, anneal the primers again. Now, in the good old days before thermal cyclers and before Taq polymerase, graduate students had to do this with buckets of water at these different temperatures. And the Taq polymerase, or the DNA polymerase, which is what Taq polymerase is, denatures at 42 degrees Celsius. And you have to boil the water to get the DNA to, so they had to keep adding Taq polymerase 30 times in a row to different tubes. Yeah, they quit. They said you can take your degree and do something impolite with it. Go to the third cycle. As you can tell, we're getting closer and closer to our target DNA. Hey, there's the red arrows. Anneal your primers. Extend your primers. Off they go. Or as I say in my class, whee! So at the cycle 4, you've gone from 1 to 8. In cycle 5, you've gone to about 21. And you can keep going, but I kind of like to do the final graph that with 1 copy, theoretically, you can end up with over 1 billion copies in an hour and a half of your target DNA. So this is basically how PCR works, polymerase chain reaction. Current slide. And I have this cool video, but I'm going to skip it because we're going to try to get out of here early. So now you have 1 billion copies of the DNA of interest. How do you see it? Again, the DNA Learning Center. We use something called gel electrophoresis, or how the freshman kids get into the lunch line before the football players do. Uh-oh. Come on. There we go. And, well, because they're smaller, so they weave in and out of the football players. And that's kind of how gel electrophoresis works. So basically, it's a matrix. You have these big molecules, and the smaller molecules are kind of serpentining their way through, and the little pieces of DNA move faster than the big pieces, so the big pieces are left behind. And a little plug for Cold Spring Harbor Labs, where I took my students for 20 years on a usually Easter Sunday. We use an agarose gel, which is agar, but it's purified agar, costs $1 a gram, and it will separate out your DNA to 10,000 nucleotides. So we can differentiate 50 nucleotides to about 10,000. Run a little DC voltage through, well, they call it a salt solution. This is the neurotoxin, trisborate EDTA. And this is now one of our protocols. When someone looks really good on paper, we actually hand them a micropipetter if they're going to work in the lab, give them a tube of blue colored dye, and say, we need you to load this well. If they can't load the well, thank you for coming in. And they're gone. And it just takes DC voltage. Because of the phosphate backbone on DNA, it has an overall negative charge. It's attracted to the positive terminal. Sometimes it tells me I have to turn on the power supply. Yeah, click on power supply. I love the animations. Here they go. So there go the freshmen. There go the football players. Oh, freshmen win. They got the fries. Okay. Putting current through? Yes. Yes. And it's kind of important that you don't touch the electrodes. Some of my students would try to bypass the safety features. We have a phrase in biology for that. It's called natural selection, which we get to show. So it turns out we stain with ethidium bromide, which is a suspected carcinogen. It's definitely a mutagen. It's a flat planar molecule that intercalates. I love that word, intercalates. It means it slips between the rings of DNA and stains them. So the bigger strand of DNA you have, the brighter the band will be. Smaller strands are a little fainter, but it is very, very sensitive, and we use gloves, and we're very, very careful. And here comes our pretty gel. Oh, it's prettier up here. But the bands run at different colors, and this is where the UV light comes on, and you can get your tan for the day on your face. And then oh, they didn't take the picture. Okay. So that's gel electrophoresis. Simple. Okay. So what do you need? Gel chambers, agarose, ethidium bromide to see the DNA, and a DC power supply. And I just like the rotating DNA helices. Notice that the bands are at different heights. That's going to be important later on. And here we are with the later on. The importance of ladders. I get to use my laser pen. So this is a ladder, and it's kind of hard to see, but the bright band here is 500 nucleotides. This is 1,000 nucleotides. Goes up to about 1,200, so you can look at this band and say, well, that's about 350 nucleotides. And it's important that you get the right number of nucleotides, otherwise you're targeting something you're not sure of what it is. Size really does matter yes. So if you're going for an amplicon that's about 900 bases, it's supposed to be here. If it's down here, we don't count it. So that's a built-in check to make sure that we're actually targeting the gene that it's supposed to be. And that's a bright one. Okay, these bright ones. This is an indicator of how much DNA you're starting with, which is an indicator of how many microbes you're starting with. We actually run a brighter the band, the more microbial DNA present to start with. And we run a serial dilution curve where we start off with 10 to the 9th CFUs, colony-forming units of E. coli, or HPV-16, or Candida albicans. Go to 10 to the 9th, 10 to the 8th, 7, 6, 5, 4, 3, 2, 1, 10 to the 0, which is 1. And I would hate to present this in a courtroom, but there is a very faint little band right there that we can see when the lights are dimmed and Barry Manilow is playing. Degraded DNA, what does it mean? Sometimes we get these bands. So all of our DNA primers are at about the same, or they're at the same concentration. So we start noticing this phenomenon. We're getting some bands that are extremely bright, and they're lower than where they're supposed to be. And it's almost like the DNA has been chewed up and shrunk. Well, it turns out that's exactly what's happened. What you're seeing here is a patient who has recognized Candida albicans, which used to be up here, and has chopped out the center of it and has had it amplified, so we're seeing this brighter band here. And I was like, well, how do you know that really happened? There's this company called GeneWiz where you can send your DNA off to be sequenced and then check it, and that's exactly what happened. The patient's immune system has gone in and has degraded this DNA, which is showing the patient's immune system has chewed up and recognized that microbe, which is kind of a nice plus. So we mentioned that. That's just a 1. It's not an active infection. And I just described that. Next. Do antibiotics interfere or affect our test? As they say in Texas. Oh, wait, Alabama. They say it somewhere. Yup. They can. How do I know this? I had the joy of discovering a few months ago that I had a crack number 18 molar. My daughter thought I was photobombing her picture because she was taking a picture of my wife and herself, and I was in the back seat going, oh, God, it hurts, right? Dad, why are you No, my tooth hurts. And I went in and found out because my tooth was cracked and had to be removed, but oral surgeons are kind of busy, it seems. So I said, wow, what a great opportunity. I keep having people ask me, do antibiotics affect your test? So I took some of our super floss, which some of you have gotten a hold of, and I flossed around that tooth that was bothering me, and I had my lab techs run it, and I didn't tell them who the patient was. And their words were, oh my God, who's this? It's me. So they put me on Keflex for 10 days. And this is kind of what happens. Here's gel 1. Nothing really showing up much here with the infection because this is Acinetobacter baumannii, which is also called Arachibacter, and Candida albicans, and maybe a little E. coli, and nothing bad. But after antibiotics, I had a bloom kind of hit. So it turns out what you're looking at is selective pressure against the microbes that we're testing for. This one, can't remember what it was, and with the antibiotic, it's gone. It just knocked them down in some cases. But if the bug was not susceptible, so this one, gone I think to here, and then this one, the very faint, I don't know if you can see it. This is what happened after 10 days of Keflex. This bug liked Keflex. It ate it for breakfast. Yeah. This made me feel better because all these, see how bright these are? That's why my tooth hurt. I had 1 billion per mil of each of those bugs. Was it? Oh yeah, it was definitely cracked. Yeah. They did suggest I could get a root canal. And I asked them, what else have you got? Well, we could extract. Emotional attachment to my teeth. What else have you got? Well, you could try Night Guard. Okay, that worked for about 6 months, and I chewed through the Night Guard. And then it was like, okay. So then they put me on doxycycline for 10 days. And then they finally got around to pulling the tooth out, and I kept the crown. It's gold. I'm going to cash it in one day. So anyway, and then this one, these little bugs right here, these are all streps. I had 5 different streps. Mitis, mutans, pyogenes, everything. Oh, and these are our positive controls. So those should not be affected. So yes, antibiotics can help or not. Basically clears the way for the bugs that are sensitive to them, knocks them out, and leaves the bugs that are, super bugs, or they just take over. And my mouth feels a whole lot better. What else interferes with PCR? EDTA detergents, so saponin, SDS, heparin, formalin. Formalin is amazing. It's a great preservative for proteins, blows DNA down to nucleotide bases, A, T, Gs, and Cs. Totally wipes it out. Chelating agents, again, the EDTA is one, and heavy metals. interfere with EDTA. I found that out actually, left out kind of an important one, hemoglobin. It's in there somewhere. Hemoglobin from blood interferes with PCR. Sorry? Oh, thank you. There it is, hemoglobin. Interferes. So when I was first setting this test up, I would take a ml of blood. It was not my blood. It was actually defibrinated sheep blood. And I would spike it with these different bugs, and then I would test it, and I was getting nothing. I was like, okay, this is, and then I read hemoglobin interferes. So I got rid of the hemoglobin, and the easiest way to get rid of hemoglobin is you just hit it with DNA-free water, lyses the red blood cells, spin it down, pour off the hemoglobin, which now looks like cherry Kool-Aid, until you have no more color, and we got beautiful results. We've actually now increased our sensitivity by using a gen-elute column, which actually purifies out specific DNAs. So, I mentioned DNA-free water. It's kind of cool. When we ran distilled water, we found out it's contaminated with E. coli DNA. Yeah. E. coli DNA is like everywhere. It is contaminating everything. So we actually had to run it through a anion and cation exchange column to pull out all negatively and positively charged molecules, re-sterilize it. We used molecular grade water. We found out that the reagents they were making, that they were sending us, the buffers and things, were contaminated with E. coli. It wasn't just our water. Questions? Yes, John.
Have you looked at silver as interference with the DNA assessment?
Not yet. It would. I know that when we played with nickel and copper and aluminum, those actually seem to stimulate the plasmid production in DNA. They liked it, except at very high levels, and then it completely wiped out. We had no bacterial growth. So, didn't test mercury. I'm pretty sure that would knock things out. Primer design. Okay, this is the cool part. Okay, I think it's the cool part. We've picked out our bugs. We've got 90. We need short 30 base sequences of DNA complementary to the forward and reverse. The picture I showed you where they were doing this kind of thing. So, I made this little animation. Double-stranded DNA is denatured. You have your primers annealing. Whee! Sticking right there. You extend the DNA in the 5' to 3' direction. PowerPoint is just amazing. Now, I couldn't get it to replicate going the other way, so I just pasted it into place. Sorry. There are limits to my technological skills. Okay. So let's say I want to make one for HPV-16. So the first thing you do is you Google HPV-16 for PCR primers. And you find highly effective detection of human papillomavirus, NCBI, NLM, NIH, also called GenBank, cited by 115. Looks promising. Can you guys read that? Good, neither can I. But I looked through, and it's kind of cool because you can go through and go, where's the DNA sequence? Find primers. Well, there's no DNA sequence in there. Some of them are proprietary. So we go to GenBank, which has all the DNA sequences of everything that's ever been sequenced around the world. And this is what GenBank's webpage looks like, www.ncbi.nlm.nih.gov. 20 years of practice, I'm pretty good at it. Click nucleotide, search HPV-16. Okay, let me read that to you. Human papillomavirus type 16, isolate HPV, BHU3, E6, which is exon 6, protein E6, and E7 genes partial. 216 base pairs. There it is. There's our sequencing, but after having taught high school for 24 years, I have serious trust issues. People lie to me. Honest, Mr. Wheeler, I turned in that homework assignment. Yeah, no. I don't believe you. I don't believe you. So we BLAST it, which is a cool acronym for Basic Local Alignment Search Tool. Oh, we have a lot of jargon in molecular genetics. And basically you take that DNA sequence and you go in and you blast it, nucleotide blast, paste it, and you get this pretty, whoa, those are wavy. They're supposed to be straight lines. And you go in and you click on one, or click on one of these bands, and it's like, oh, I found the entire sequence for HPV-16. Click the accession. Starts at 562, amplicon size 296. Here's the entire sequence, well, part of it for HPV-16. Oh, this is a small one. The one I'm looking at right now is P53, which is a human tumor suppressor DNA fingerprinting. It's 20,600 bases. You just scroll down nucleotide. It's exciting. Well, kind of exciting. So here's the sequence that we wanted, 296 bases. It's a nice size for an amplicon. We have the template, we have the primer. And it's kind of cool if you order in the afternoon, because it takes about that long to find this, and say, okay, I want this sequence of DNA from Sigma Genesis, also a subsidiary of Sigma Chemical Company. You will have your lyophilized FedEx shipped in about 2 days. So they're very quick. You do want to make sure you get the primer sequence right. And that's kind of what their web page looks like. And then this is just a recipe of AdTac, buffer DNTPs. Oh, DNTPs, deoxynucleotide phosphate, triphosphates. This is ATP, GTP, CTP, TTP. They're the building blocks for DNA, high energy molecules. They're the most unstable things we can have. We like to store them actually at about minus 70 degrees right here. And then molecular grade water, again, or DNA-free water. And they found the TAC, by the way, from Thermophilus aquaticus from Yellowstone National Park in the geysers. They found the bacteria. It likes it at 100 degrees Celsius and above. The DNA polymerase is stable, and hence it's called TAC polymerase. A little history. Only takes 3 microliters of sample per well, so when you send us a drop of blood, that's plenty. That's all we need is 1 drop of blood, 1 tooth. We love getting infected root. Well, okay, love is probably. We enjoy getting the infected root canal teeth because they're easy to purify. You basically put them in a little tube with some DNA-free water, vortex like crazy, which pulls the DNA bugs out. And jump. Yes. Yeah. You can send us a survey, so you can actually go in and floss all around the mouth, or you can do like I did and pick the 1 tooth that's bothering me, or you could use dental points. I don't know about you guys, but I find that patients salivate a lot, and they make the dental points kind of soggy, and they're kind of hard to get to. We're finding the super floss works better, actually, than the dental points do, and it's a whole lot easier to do. So, and then we inoculate in the 3 microliters of samples. So since we're running about 100 different tubes, we use 300 microliters, which is 0.3 of a mil of sample. We usually have enough to run it a couple of times. Mix it all together with this PCR buffer magic mix. Put it in a thermal cycler, which raises the temperature, lowers it, raises it a little, raises it to boiling, cycles through 30 to 40 times, depending on what our control is, or what our program is. Makes 1 billion copies of your DNA. How do you know if the primers are detecting your target DNA? Well, we have found it extremely difficult to grow some of these bugs, like the HPV 16, 18, 58, 52. Okay, we're looking at 14 HPVs right now. So what we do is we use a company called GeneArt, a subsidiary of Invitrogen Life Technologies, who actually will say, I want this DNA sequence. They will clone it into E. coli, and your E. coli will now make the DNA of your choice. Now, When I first did this, I got a little biosecurity alarm from Germany, who was making this for me, because I had put in the toxin for Clostridium botulinum. Yeah, and they basically asked me, "Are you a terrorist?" Well, yeah, they actually had me sign a form which said, "I promise I am not a terrorist, a bioterrorist." No, I'm serious. I signed a form which I'm not a bioterrorist. I had to have proof that we actually were a real lab, and I promised not to share with others. No, really, I'm not allowed to share this with others. They get real testy about that. And we put it in E. coli because, frankly, if you can't grow E. coli, there is something wrong with your technique. I mean, everybody can grow. Though, you know, a lot of people when they make bread for the first time don't realize yeast is a living organism. And they say, warm water. Well, I'm in a hurry, so if I put it in hot water, it'll grow faster. Yeah, they all die. And your bread does not rise. Not for a long time. Beauty of the primer design is you can say, I want an amplicon between 200 and 1,000 base pairs, so it's not confused with any possible primer dimer, which is about 100 base pairs. And questions? No? No. Well, actually, I hear there is one tomorrow morning. So this is kind of what our positive controls look like, and you can see that they're all at all different kinds of heights. This is our ladder, 100, 200, 300, 400, 500, 6, 7, 8. Okay, they compress a little bit. So we're having our positive controls, and they're supposed to be at a certain height, a certain size. If they're not there, it's wrong. And there's something wrong. Fortunately, it's always been right. Sometimes we get two bands, which Leslie, my molecular geneticist, found out that in, I think it was Ehrlichia, that the sequence repeats within its own sequence, which I hadn't realized. She said, "Did you do that on purpose?" And I responded, "Yeah, of course." So, for example, cytomegalovirus, 388 bases, actinomyces israeliae, 264, Clostridium difficile, 960, HPV 16, 297, Pseudomonas aeruginosa, 726, EBV. Okay, so we know that we're hitting the exact right target. If it doesn't hit, it's like, we're picking up a cousin. Generate a report. Clostridium botulinum trumps E. coli. So if you get Clostridium botulinum, you will actually get an email or phone call from me saying, "Hey, how's your patient doing?" Because they just tested for a neurotoxin we only find in Clostridium botulinum. We also go an indicator of, okay, if you only have 10 of these microbes, it's not a really big deal. If it's E. coli or something, Clostridium tetani, we get a little more excited. But we also ask, how's the patient feeling? You know, a lot of times the tooth has been extracted and they're actually feeling better. This is an example of our report. This is what a lot of people like, this bar graph coming out, but now we've had some feedback from people who say, I would just like a list of the microbes and then color-coded with like about how much are there. But then you have the report and it's like, okay, so as Dr. Margolis asked me, now what? Well, you could make it someone else's problem and say, so I think you should go to a naturopath, you should go to an infectious disease fellow, you know, and maybe they can help you. Or let me explain why we're not a diagnostic lab. We don't have an MD on staff. That was the only thing that would change. We just don't have an MD on staff. Clinical significance, number one, whether microbes are of clinical significance. Find the antimicrobial that targets these microbes or another treatment. Send a patient to an infectious disease fellow who will sometimes tell you, well, the mouth is full of microbes anyway. And it's like, well, they're not wrong. It's just usually if you have tetanus or gas gangrene or botulism or something in your mouth that, that's not considered normal flora. Or we actually found anthrax toxin once, which is found in dirt, by the way. So what do? Well, I skipped a step. Dang it. Did I? Yes, I did. So in our report, which of course I don't have a picture of, but you can see it online, we actually have text sheets which are currently being updated, which give a description of each of the bugs. What does it do? Like HPV-16 can be an STD. They're actually finding evidence now that it doesn't absolutely have to be an STD, that HPV is destroyed by bleach, fire, and autoclaving. Purell hand wipes, or Purell, not so much. Glutaraldehyde, not so much. But the others work really well for disinfecting. So kind of gives you an out. I had a patient call once and asked me, so I'm tested positive for HPV-18. Is that, can that be sexually transmitted? Yes, it can be, but thank you, click. And I thought, there's going to be a very interesting conversation tonight at home. I wish she had let me finish and say it doesn't necessarily have to be. It's a virus. All warts are caused by an HPV, and all warts, so it's transmitted. Anyway, we do have a list of what the CDC recommends for treatment. Leslie is working on an updated current literature review for the new treatments because these bugs are sharing genetic information a lot. Bacterial conjugation, they're transferring resistance transfer factors. They're becoming more and more antibiotic resistant and getting harder and harder to treat. So we have found that CDC recommendations are already out of date, even though they're 3 months old since we made the new ones. We have this wonderful thing called the internet, and people are using it. And they are going to be coming to you and telling you things you may not know. And you need to realize that there are a lot of websites that have some questionable data, and we'd like you to have several options. Refer your patient to a naturopath is another way to go. But this part, listen to what they have to say, as they may have discovered a new treatment or paper with valuable information. Let me just guess. Show of hands. Anybody have that happen? 1, 2, 3. Oh, that your patient has come in and has actually known more about their disease than you do and possible treatments. The reason for that is very simple. They have a vested interest. They're sick. They want you to help them, and they may have spent a little more time than you've had to research it than they have. They've had more time. So, questions? Yes, in back. We have done 2, and they're not infected, but there's a biofilm on the outside that is there, and we're finding strep mutans, which is causing tooth decay. Most of our tests, the closest thing we have to normal flora is strep mutans. Everything else, strep pyogenes, they're virulent pathogenic, yeah. And usually when we get these, the people are not feeling well and the tooth is bothering them or something's bothering them. So we're kind of being selected for things that are causing problems. And we do have some like mine that just light up the panel. Why not treat with antibiotics preemptively? Well, sometimes it's not a bacteria. Sometimes it's a parasite like entamoeba. my daughter was exposed to Giardia in Rocky Mountain National Park. She had rinsed off her hands to cool them off in the fountain. She was 6 years old, 5 or 6 years old. Stuck her thumb in her mouth. She got Giardia from that. Didn't drink the water, just stuck her thumb in the mouth. That was all it took. Took them 6 months to diagnose. It finally took a head of pediatrics, right as my daughter stuck her thumb in her mouth, to figure out that's what she had. So she was on multiple antibiotics, no effect. Put her on an antiparasitic, and she was cured. I mean, so what I'm saying is you want to treat the patient for what they have. You don't want to treat them for something that they don't have. What if it's Epstein-Barr, HPV, Candida? Antibiotics aren't really going to affect those. Pseudomonas aeruginosa. When I worked at Shriners Burns Institute in Cincinnati, we would do Kirby Bauer and Mueller-Hinton plates, which are you streak the plate with Pseudomonas, you drop 16 different antibiotics on it, you look at it the next morning, 2 of the antibiotics work. So you prescribe one of those 2 antibiotics, or tell the docs to prescribe that. A week later is eating those antibiotics. So we're kind of losing the war on antibiotics. What else do we look at? Line panel. This is becoming ever more popular. These are the different things that we're looking at. Borrelia burgdorferi. So the flagellin gene, F7 for Borrelia, is the gene that makes up the flagellar protein that allows it to corkscrew in. And it's fairly specific. It blasted well, but University of Australia said, yeah, but it might react with other things. So then we looked at outer surface protein A, B, and C. And when we get a match with those, we're pretty sure that's what it is. We also look at Borrelia miyamotoi, Borrelia recurrentis, co-infectors, Babesia, Bartonella, Ehrlichia, and our newest one, Anaplasma phagocytophilium, which, well, that's an interesting one, parasites. We also test for ApoE. Dr. Huggins was of the belief that if you ran a genetic test, you needed blood. You don't. You need cheek cells. You get 1,000 times more DNA from cheek cells. And let me tell you, after having stuck my fingers several times for a drop of blood, cheek cells are definitely the way to go. You can do a swab. You can rinse your mouth out with saline. Saline gives about 10 times more DNA, but it's a little more difficult to transport. This gives you all this really cool information that would be in the report for ApoE. Okay, 22, 23, 24, 23, 24, 33, 34, 44. Okay, and it's kind of a balance, because if you're a 22, you have very low risk for early onset of Alzheimer's, but you're at very high risk for heart attack because your cholesterol is in the 800 range or higher. The average population, 70% is a 3-3, and if you're a 4-4, early onset of Alzheimer's, but your cholesterol is low. Or, well, that's kind of the Reader's Digest condensed version, which I just gave you. I'm a 3-3. I always test myself first because I'm always carrying my DNA around. Hardy-Weinberg equilibrium, I don't know if anyone remembers that or not. Shows no selective pressure. There's definitely selective pressure for a 3-3. Improving our test, best part of my job or position. ApoE, cysteine, ApoE2, cysteine, ApoE4, arginine, ApoE3, cysteine, arginine. And then my question became, why can't you have an arginine forward and a cysteine reverse, the opposite? And the answer is you can. No one's done it before, except me. So I like to be number one. So now we actually are looking at ApoE2, 3A, 3B, and 4. All 4 possible genotypes.
Is there a difference between AP3AB in the sense of being able to remove the mercury from your body?
No, it's just really cool.
I didn't.
No, it should be exactly the same. Should be. We actually haven't found anyone with this yet, but we don't get a whole lot of requests for ApoE. But this is how you read an ApoE gel. Is pretty pretty easy. Well, I'm doing okay. Okay, so this is before I put in the 3A and 3B. So we're in groups of 3 per patient. This line on top is actually a DRB, as a tissue typing marker, which we just showed that the PCR is mixing or is working, but it's also acting as kind of a sponge for excess reagents. So this column is 2, 3, 4. That's me. I'm a 3-3. That's how easy it is to read these. I feel almost guilty taking your money. It's that easy to read. Of course, the techs disagree with me because they have to set up the test. But this is a 3-3. This is a 3-4 because 2, 3, 4. Ladder, 3-3, 3-3, 3-3, 3-3. Faint, 3-3. Bad DNA prep. 2-4, 3-4, 2-3, 1, 2, 3, 3-3, 2-4. That's how easy it is to read a test. Oh, by the way, when you give people these results, you should refer them to a genetic counselor. That's what got 23andMe in trouble. They had no genetic counselors. Questions? Actually, if you Google genetic counselors, they list them by state. Whoever asked that question.
Is the way you are genetic testing pretty similar to metametric or genome testing? Genobio lab?
A Genobio lab?
No, Genobio laboratory. Metametrics. They are pretty well known.
Oh man. I don't know because I haven't. If they're testing using literature like I did, yes. If they use GenBank, you go in and find the sequence and do it. So it should be the same. I mean, there's pretty much only one way you can test for ApoE 2, 3, and 4.
No, I'm talking about microbiology DNA test.
Oh, I don't know. I don't know. They keep me in the lab doing research. I don't get out much.
So another question is there any different testing the DNA component?
Oh, who else does DNA testing for microbes? Not for PCR. Johns Hopkins is doing it. And I think the Mayo Clinic is doing PCR testing for microbes. Is that what you're asking? The DNA, yes. Okay. Okay. So you're asking, oh, the same techniques. They could be using regular PCR. They could be using something called real-time PCR, where they're looking at a DNA probe with a fluorescent tag on it, which shows how much is there to start with. The problem with that kind of testing is you can only test for one microbe at a time. With our technique, we look at 90. Okay. So they could be. There's another company called Pathogen, which uses shotgun genomics, which uses nonspecific primers. We actually sent spiked blood samples off to them, 43 known bugs run in a double-blind experiment. They missed 24. So, yes, other companies are doing it. I don't know if they have the controls in place. They must have the controls in place that we're using, I would think. I don't know if see me after, we'll talk. Okay. Gluten intolerance test, beautiful slide, very complicated. Shows basically HLA tissue typing for DQ2 and DQ8, which are the abbreviations for in a autoimmune response where these markers actually are DQ2s and 8s. It's an autoimmune disorder, makes IgA antibody, targets the protein, wheat protein gliadin. 95% predictive for sensitivity or intolerance to gluten. And the samples for testing, again, are cheek cells because you get 1,000 times more DNA. The story behind this is there was a workshop Dr. Huggins was running, and everyone seemed to be interested in getting a gluten intolerance test. And I had just developed it 2 weeks before, and it seemed to be working very well. But I had everyone's cheek cell samples, and I ran it, and everyone in the workshop had a DQ8 exon 3. So my first thought was, damn, I thought this test worked. I'm sure it worked. And so I mentioned Dr. Huggins. I said, I'm sorry, I don't think the test is working. Everyone in the workshop that I tested, except for the negative control, had a positive result. And Dr. Huggins looked at me and smiled and said, Bob, it's a gluten intolerance workshop. Everyone is gluten intolerant. And I looked at him and said, you might have mentioned it to me. It would have been nice to know. I thought I was going to have to throw out the test and start all over again. So it is 95% predictive. There are different ways people manifest. Some people have the gut-wrenching intestinal diarrhea. Some people have their sinuses fill with fluids. It's an allergic response. And like I said, 95% predictive. This is our negative test. Oh, it's a beautiful ladder. If nothing else, you always get a beautiful ladder. Okay? And this is the positive test, where these people had a gluten intolerance right at about 296 bases where it was supposed to be. So improving our gluten intolerance test, I found some of the exons are so small that they wouldn't show up. They're only about 50 nucleotides long. So what I did was I went on either side of the DNA sequence into the intronic sequence and made them about 150 to 200 bases. So we expanded from 4 nucleotide, 4 exons to 8. So we have a bit of a more sensitive test. Questions? By the way, I'm gluten tolerant. Tested my DNA. Viral panel. This is one no one has ordered yet, and I've always been a little surprised. We're looking at 25 different viruses: adenovirus, cytomegalo, Epstein-Barr, varicella zoster, herpes simplex 1 and 2, the 14 different HPVs that cause cancer, hepatitis A, B, C, E, and G. Hepatitis A, C, E, and G are RNA viruses, and we actually have to use an enzyme called reverse transcriptase, which takes the RNA back to DNA and then use PCR on it, but we've worked it out pretty well. But it's labor intensive, so Leslie has kind of harvested my viral panel and put a lot of these viruses now into the full view, which we run routinely, which I think I just described there. And on the, oh man, yes. On the horizon, I'm currently working on a hereditary cancer panel, BRCA1, BRCA2, give the guys fair time, hereditary prostate cancer. We don't get away scot-free. P53 is the one I'm playing with right now. Is kind of a cool gene. About 20 years ago, it became Time Magazine's Molecule of the Year, which I didn't even know they had a Molecule of the Year. I've heard of the Man of the Year, the Woman, no, but P53 is a really neat gene, does a few things. One is this a proofreader for DNA mistakes. It's also a tumor suppressor gene, 20,600 bases, I think. 10 exons. It's the one that when you get sunburned and you peel, that's your P53 gene kicking in. Because your DNA is so damaged that the P53 gene can't proofread it and repair it fast enough, and it sends a signal to the nucleus and tells the nucleus to tell the cell commit suicide. It's called apoptosis. jargon. Love it. And that's why you peel. And if you don't peel, you might develop a little something called melanoma. So, and there's also hereditary skin cancer. That's one of the genes. I can't remember. It's one of these. If you are one of my students, Daisy Snyder, her research project in my DNA science class was looking up this hereditary skin cancer, and she decided to throw in ethnicity. And it was real interesting. African Americans, 2% had the gene. Hispanics, 4%. Asians, 6%. Caucasians, 31%. Unless you're of Irish or Scottish descent, 68% have the gene. Okay? So keep that sunscreen coming and wear a hat. Wear a hat. So we're kind of playing with that. Von Hippel-Lindau, TP53 is really P53. So we're playing with those. Final chance for questions. Yes, Joan. Oh, cheek cells. Oh, I'm sorry. For viral? Anything with DNA in it. Blood would work. An open wound would work. A tooth would work. Cheek cells will work. For the cancer panel, it will be cheek cells. One last.
When you take DNA from a tooth, how do you extract it?
Well, we used to take a hammer, basically, and break the tooth into little bits and then extract the DNA. We actually ran that through a test where we take the DNA, we put it in a large tube with an extraction medium, vortex like crazy. We now send it through a gene elute column, which purifies out the DNA and separates out all the interfering substances. So works pretty well. Yes, way in back. John.
What progress are you making toward parasite identification using your technology?
Well, we're looking at entamoeba, but I want to look at Giardia. And here's the thing. That's a very good question. We are flexible. If you want us to look at something, send me an email, and we should have it online within a month. If GenBank has the DNA sequence. If they don't have the sequence, we can't do it. Yes? Uh-huh. It kind of depends on how vigorous you are with your flossing. When I was doing my tooth, it really couldn't hurt it any more than it already was. So I was drawing blood. So I was getting a little clavicular, and I've seen some, I don't know about your dental hygienist, but they're fairly vigorous with my teeth. So to answer your question, it could be some contamination, but you could use the dental point if you want. We still put those out to chew on a popsicle stick and cause it to ooze up. Like I said, the problem with it is it gets kind of soggy. Yes? Uh-huh. I'm sorry, if they were what? For detecting them, no, because we're seeing the DNA. We're not looking at the cell wall. No, it doesn't matter at all. In fact, we have people say, well, but when you pull out the obligate anaerobes, doesn't it kill the bug? And it's like, we don't care. DNA is still there. Oh, I left that slide out. Dang it. Ozone. People have asked about ozone. Ozone is very interesting. Ozone breaks down into oxygen. It's very deadly to obligate anaerobes. It kills Clostridia beautifully. Clostridia is the worst of the bugs we're looking at, really. Wipes them out completely. The bad news is that when you knock out the obligate anaerobes, the facultative anaerobes like E. coli bloom. They actually burst into population, and you can get a burst. If I had a choice between tetanus, botulism, gas gangrene, and E. coli, I choose E. coli, please. Okay.
Are there any more questions?
I have one more slide.
You said final chance.
I know, for questions. On my horizon. Okay. I have something to share with you that's kind of important. I just found this out about an hour and a half ago. This is Turks and Caicos. It's our favorite place in the Caribbean, British West Indies. People are very nice. That's my wife and I. Actually, this is in Kauai. I promised her for 27 years I would take her to Hawaii. I'm a little slow. I got there. Took me 27 years. This is my daughter Jennifer, who got married June 6th of this year, and she told me I'm allowed to share. She's making me a grandfather.
Congratulations. Dory, why don't you give the thing and then I'd do a presentation.
All right, thank you. Okay, the code for this session is BW, as in Bob Wheeler, BW 1010. So that's BW 1010. And then please bring all of your sheets, put them in your envelope, and bring them to my booth at the Talk International booth in the vendor area. And just drop everything off, and you'll get your certificate back in the mail within about a month. If you're an AGD member, I'll be reporting that directly to AGD for you, plus you'll get your certificate in the mail.
Okay, a few things. First, Bob, thank you very, very much. I don't know how to thank you for all the work that we've done together over the years and how everything has gone. I have no idea what's in these crazy